Welcome to the session. I'm sorry about that. I'm going to talk today about the interaction between the nervous system and the penile vascular system. Do I point? Where do I point?
There. No, towards the back of the room.
Oh, the back of the room.
You were
Okay.
supposed to have 10 minutes. I don't know.
10 minutes. Okay, this is easy because I had it scheduled for 5. All right, there we go. These are my disclosures. Nothing is relevant to anything I'm talking about today in this talk. So this is my outline. Very basic understanding of both neuronal and vascular control of erection, and then a little bit about the anatomy of the penis, the arterial supply, the tunica abyggenia, and the corpora cavernosa or corpus cavernosums. What I'm not going to be focusing on, because my colleagues over there are going to be doing all this, is talking about the pathobiology of erectile dysfunction or a comprehensive overview of signal transduction. So I'm not going to talk about that in detail. I will bring it up as it relates to historical understanding of the vascular control and neuronal control of erection. So erection biology is a neurovascular event. Clearly, we know this after everything we've heard this morning. It depends on 4 intact sort of organ function, which is neural integrity, functional penile vascular system, intact androgens are hormones, in particular testosterone, and healthy corporal tissue or structural integrity. If you have any impairment in any one of these 4, then this may lead to quote-unquote erectile dysfunction. So normal erectile function involves 3 synergistic and simultaneous processes, which is neurologically mediated increase in penile arterial inflow, relaxation of the corpora, and restriction of venous outflow from the penis. And that's what I'm going to spend the next kind of 5 or 6 slides reviewing. So first, what about the innervation of the penis? Dr. Walsh, actually started in the 70s. Ultimately, this is just one of his papers that looked at the innervation of the penis, once again, coming from the pelvic plexus, S2, S3, S4. I'm not going to be talking about the centrally mediated effects down the spinal cord. I don't think that necessarily is relevant for what we're going to talk about with the end organ, at least right now. S2, S4, parasympathetic, sympathetic innervation, the neurovascular bundle are the quote-unquote cavernous nerves run lateral to the prostate. It also innervates the bladder, the pelvic floor, the urinary rhabdosphincter, and as well as prostatic function. So this is sort of our understanding of what of how nerves, how the penis is neurologically innervated. Arterial supply, once again, basic, coming from the internal pudendal artery. This is relevant because when we talk about the penile vascular system, we have to be able to delineate between the arterial system as well as the end organ corporal endothelial small arterial system. So the main blood flow to the penis is coming from the internal pudendal artery. We heard from our last speaker how if this is impaired, you can result in impairments in corporal smooth muscle relaxation or contraction. The venous system, once again, same process here, which is relevant for the having a sustained erection. So this is the flaccid state. You have your cavernous artery, which is that tributary from the internal pudendal artery, where it leads to increases in blood flow in the corpora cavernosa, which is a sinusoid of smooth muscle, endothelial cells, collagen, elastin, and this is where you heard a lot from our previous talks about the effects on fibrosis and what occurs. In the flaccid state or non-erect state, those veins, which you see up top, which is this emissary veins, are just relaxed. But when you have an engorgement, you see an increase in those veins where you get essentially a compartment syndrome, where you have an increase in blood flow. And if you have an intact tunica abyggenia, if you have a venous system which is also intact and not compromised, you get this closure of the veins and you get this rigid erection. And this is where you hear a lot of research revolves around being able to determine factors which influence the corpora cavernosa in its ability to relax, its ability to have smooth muscle integrity and content, endothelial cell content, and how that affects this increase in corpora cavernosa. Now, the tunica albuginea, I think, is something that we probably we don't talk as much about as it relates to erection physiology. Obviously, in Peyronie's disease, it's discussed a ton. This is Jerry Brock's paper from the late 90s, where he showed how the tunica albuginea, how it has these sort of these pillars that run through the corpora cavernosa. And the tunica albuginea, if there is any type of atrophy, accompanies things like diabetes, for example. If you have a loss in the collagen there, or if you have fibrosis or an increase in the tunica albuginea, such as in Peyronie's disease, this compromises the erection process, and you can get what's called veno-occlusive dysfunction. So this is actually an old tracing from, I believe it's a monkey from Tom Liu in the late 80s, where he was looking at arterial flow through the internal pudendal artery, and you see the different phases, which I'll describe in detail, where you have this increase in the internal pudendal artery inflow. You then see this increase in the corpora cavernosal pressure, which is sustained until, in this case, it was a stimulator, but after orgasm, then the erection comes down. So if you look at the phases of erectile cycle, and this is, I'm purposely talking about sort of the basics and what we understood maybe 20 years ago, 25 years ago. In the flaccid state, this is predominantly governed by the sympathetic tone, so increase in norepinephrine and the like. This, during filling phase, is where you develop this parasympathetic stimulation, things like acetylcholine, the non-adrenergic, non-cholinergic innervation, where you see this increase in arterial flow. But in order for the corpora to increase, you actually need the inflow from the internal pudendal artery. Once again, oftentimes studied, but we don't talk about it as much as I think we should. During the tumescence phase, which is the rigidity, this is when you see that those veins pushing in the subtunical space, you get your full erection, and importantly, the increase in intracavernosal pressure is about 90% of your systolic blood pressure. So you really do get increases in the pressure. So if your blood pressure drops in response to some vasoactive agent, your ability to get that increase in corporal pressure is also going to be reduced. During detumescence, this is once again governed by sympathetic stimulation, and then in sympathetic tone. So I keep on banging on about this, but as it relates to arterial inflow into the penis, it's all dependent on resistance. So resistance is important through the IPA, or the internal pudendal artery, as well as downstream effects on the actual corporal bed. So if you look at pudendal artery resistance, this is work that was done by Mike Adams, Joanna Hanna in Canada. What they showed was that 70% of the total resistance of the entire penile vasculature, so remember the entire penile vasculature includes the internal pudendal artery all the way down through coming from the internal hypogastric artery down through into the crus, so the base of the penis, and then into the corpora. And that the majority of resistance actually occurs in the internal pudendal artery, once again highlighting the importance of arterial inflow. And if you look at what happens in the internal pudendal artery in patients that have different pathologies, you see that, and this is in a lower panel, this is actually cadaver studies done by Joanna Hanna many years ago, you can see that there is a compromise if that patient has atherosclerosis, smoking, hyperlipidemia, all risk factors for cardiac disease. You can see that there's a narrowing in lumen there, which reduces arterial inflow. But really, when we think about which you just heard the first session on, and I suspect my colleagues are going to talk a lot about, is the actual corpora cavernosa, or the corporal bed. And Mikkel in Prague in 1973 made this comment that erectile dysfunction is related to diseases of the vascular bed. I think he meant the internal pudendal artery, the common iliac artery, but when we think about it in the research we're doing, we're actually talking about the end organ penis. But these are things that need to be studied simultaneously. So as it relates to scientific discovery, the field was like blown away and kind of really became what we are today in the early 1990s. In 1992 was Lou Ignaro's description of nitric oxide binding to guanylate cyclase and increasing cyclic GMP in multiple vessels, not necessarily the corpora. He later showed it in the corpora cavernosa. And Moncada showed in 87 the role of endothelial-derived relaxing factor, EDRF, back then, now known to be nitric oxide. The Nobel Prize was ultimately given to Ignero for this work. So this is, when this discovery was made, it sort of led to the production and understanding of the vital role of nitric oxide. So 2 investigators on each side of this country, one on the West Coast, one on the East Coast, Bud Burnett at Johns Hopkins, Jake Rafer and Nestor Gonzalez-Cadavid, and Luke Ignero made this discovery almost simultaneously within a couple of months of each other. So this is how we understand the main physiological mediator, which I don't need to go into because we just had a whole talk about that. But as it relates to regulation of resistance, in order for the penis to either contract or relax, you have a number of different factors, signaling factors that contribute to this. And it's a vascular balance. So if you have more vasodilation, it tips to where you get more arterial inflow. And if you have an intact structural integrity, you get an erection. If you have intact hormones, you get an erection. And if you have more on the vasoconstrictor side, you will result in detumescence or less erection. So once again, broad strokes. This slide is just to make the point that there are numbers of signaling pathways that have been shown to be involved in this process, coming from the endothelial cells, from the nerves that are innervating the penis, as well as factors downstream in the smooth muscle that result in relaxation and contraction. My colleague who's sitting 2 seats to me, Clinton Webb with Kanchan Chitterley, and was it 2002 or 2001? Do you remember? 2001. 2001, yeah, thank you. Nature Medicine paper described this, which is the importance of, for the first time, in my opinion, we saw a signaling pathway that was important for vasoconstriction, where if you inhibit, you can actually cause a really robust erection, and that's in the Rho kinase system. Rho-A is upregulated in things like hypoxia, diabetes, hypertension, and this leads to more downstream effects in the corporal bed, such as fibrosis, and ultimately leads to a more vasoconstricted state. And if you inhibit Rho kinase signaling, Rho-A or either Rho kinase itself, you get vasodilation. This is ultimately further described that this can lead to changes in other pathways, such as the nitric oxide, cyclic GMP, PKG point. So I'll end there and just simply say that hopefully I've provided you with some basics of where the field is right now, and I'm hopeful that my colleagues can now fit in where we see a lot of the pathobiology related to what will govern, hopefully, ED treatment in the future. Thank you.